Right, great. Thanks for the invitation. Sorry we're not seeing each other in person. I was really looking forward to it, but I totally understand that it makes sense to keep it online again, hopefully for the last time, and hopefully we'll see each other next year, all in person. So yeah, I'm gonna move it forward a little bit from the pediatric presentation of this disease to the adult form. And as you all know, the adult form affects, in fact, the majority of individuals affected by this disease. So just quickly, these are my disclosures. The most important disclosure being that the work I'm presenting is work done by several other people who are, I think, all online, and they can talk much better than me and are much smarter than me. So I appreciate the fact that I'm presenting here. So AMN, the adult form of ALD in males is something that starts somewhere in the 20s. There's rare occasions that, like a late adolescent, teenagers may have some symptoms. Also seen individuals presenting as late as in their 60s with the very first symptoms. So it's really a huge range in time of onset. And it's from a pathological perspective, a quite different disease than childhood cerebral ALD. It's primarily located in the spinal cord and the long tracts that are in the spinal cord that connect essentially the brain with the rest of the body. And so the tracts that are involved, we are primarily the sensory tracts called the dorsal column here in the back of the spinal cord. You can see it here, this area that's demyelinated, and the lateral corticospinal tract. So these are the motor fibers that go down and the sensory fibers that go up. And so it results in a mixed picture of sensory issues that result in at times in pain and tingling and numbness, but at the same time also can affect significantly gait and balance. And it can affect motor function in terms of causing weakness of the muscles and also stiffness in the muscles. And there's also pretty prominent bladder, sexual, and bowel dysfunction in this disease. And as you know, unfortunately, a good percentage of these individuals can also develop an inflammatory disease in the brain, similar to children. And so, given that it's the most prevalent form of ALD and affects men and women, there has been a great emphasis on trying to figure out a therapeutic. And the biggest sort of obstruction in identifying therapies is not that we don't have enough therapies. In fact, there is now, I think, around 10 companies that have a clinical trial or clinical research development program for AMN, but it's such a slowly progressive disease. So the good news here is it's a very slowly progressive disease. The bad news is that it's so slowly progressive that it's very hard to measure a change, and that's been a challenge for many years. And this is a publication when Dr. Van Giel from Netherlands came to visit Jay Raymond and Hugo Moser and looked at the AMN cohort at Kennedy Krieger back then, and they looked at 60 AMN patients that they called sort of pure AMN. So and then over a period of 16 years, they developed a moderate handicap. And when you look at this actual data here, they used the neurological score, and you see that this score from baseline to 16 years plus minus 9 years. So at the minimum, somewhere around 7 years after baseline, had only progressed by 1 point. And so that makes it incredibly difficult to assess the effectiveness of a drug trial if it takes so long to have a notable neurological worsening that you can assess on an exam. And therefore, there's been sort of a quest for markers that can be more quantitative and change more rapidly. A study published a couple of years ago by Mark Engelen's group, by Irene Hofnagel, who has come to this meeting before, basically found similar results. When you look within a 2-year period and look at the common neurological scales, one is called EDSS, the other one is called SS-PROM. This EDSS is a very commonly used neurological scale that we use in the clinic for multiple sclerosis, AMN, many other neurologic diseases. Basically, there isn't really much of a change within a couple of years. makes it very difficult to again use them as an outcome measure if you want to do a clinical trial when you only want to treat somebody for a short amount of time and see if the drug is helping. You notice here, though, that the 6-minute walk test did change, although it's a small percentage of change, but there was a change there within a couple of years. And so we have really had over the last 20 years or so within our group at the Kennedy Krieger really focus on trying to figure out quantitative measures of outcome. And so there's different outcome measures. The group that we talk the most is quantitative performance measures. This is a term that the FDA uses that are sort of considered by the FDA typically as valid outcomes, although they still have to be shown to be meaningful to the patient, because the FDA doesn't care just really about numbers. They want to know if those numbers are meaningful. So here is the sort of the one that's currently accepted by the FDA for AMN and was used as a primary outcome measure for a recent clinical trial and is used in many, many other diseases. And that's called a 6-minute walk test. And there's a couple issues with this test. For one, it's a walk test. And so walking is motivation dependent, maybe depending on many things, how much pain you have, what the weather is like that day, what you had for lunch. And then also there's a big age effect. Of course, the older we get, the slower we get. Anybody who runs here will know that. But when you do that and you look at men and women, and this is a result of a clinical trial that was done with Lorenz's oil about a decade ago or more now. And so we just published these results finally. If you look at women and men, you see that in women, really over the 2-year period, there wasn't really a change at all, while in men, there was a decrease. And so at least there is a decrease, but again, it's not the greatest outcome measure, especially in women. It doesn't seem to be changing much within the time period. So we have a motion analysis lab at Kennedy Krieger, which is run by our Chief Science Officer, Amy Bastian. And there's been a group of people working there now for many years on AMN. There was a person who left the institute called Dr. Zakowski, Kathy Zakowski, and then Jen Keller, who've been working together for many years on AMN, and more recently, Dr. Smith-Fine. And I think Jen and Amina are both here today, so they can correct me if I say something wrong. But this is how a motion analysis lab looks like. There's several of these labs in various academic institutions, and it's basically a group of people who really quantitate how we walk, how strong we are, how we sense things, using quite advanced sort of robotic technology and a lot of machine learning tools to analyze how we do things in motion. And so one of the tests that in the early 2000s seemed to be very helpful in AMN was a test that would assess how much we sway when we stand. And so it's called a sway amplitude or a sway test. And so basically, the dorsal column in AMN is the pathway that carries the information to the brain in terms of where we stand. So if the dorsal column is affected, we get what we call a sensory ataxia, imbalance, because our feet do not feel where we are in space. And it gets much worse when you close your eyes, because your eyes can still help you, guide you in space. But when you close your eyes, that signal goes away, the visual input. And so it gets really much worse. And so one of the first observations was that when you stand on this force plate and you basically have a plate here that can measure exactly how much you move. So the slightest motion here can be measured by this plate. And you can draw out a line here, how much you sway, and you can see how that moves in a control. And this is an AMN man. And then we looked at this amplitude in terms of change from the midline, and we see that there is a correlation with the neurologic disability. This was noted back then. And it was noted back then also that when you have the same subject, an AMN patient here with eyes open, as soon as they close their eyes, it gets a lot worse, which is something that many people experience when they take a shower and so on. They say, when they close their eyes because they're washing their hair, they totally lose their balance. And so we were very hopeful that this might be a good quantitative and sensitive measure of change. And again, the group of Mark Engelen in the Netherlands and Stefan Kemp, they've been looking at this more recently in a larger sample size in a Dutch group of patients. So So it's not just an American thing. This is international. And then these terms are like eyes closed, feet apart, eyes closed, feet together, eyes open, feet apart. And bottom line is we see that male AMN patients are off all across the board. And again, they saw same observation as we did, that it correlates quite well with the neurologic disability score, and it inversely correlates with your walking speed, which makes sense. So the faster you walk, the better you sway. The slower you walk, the worse is your sway, which the worse is your balance. So that kind of makes sense that these things go along with each other. The big question has been, okay, about how much does that change over the course of time? And again, as I mentioned, there was a clinical trial with Lorenz's oil that was done about 15 years ago by Jay Raymond and Hugo Moser, and it actually was terminated prematurely because of the side effects in the placebo group and some recruitment issues. But the data was there in the placebo group that we could analyze to see, how these variables changed. And this is a paper that just came out recently. So these are the age groups. So we had a pretty good number of individuals at baseline. Unfortunately, a lot of them dropped out over the 24 months period, but we still had a good number of men and women after 24 months that we could assess. And so when you look at these individuals, you see here with the eyes open, feet apart, eyes open, feet together, with both of these, the men are blue, the women are red, as opposed to the walking speed where only the men got worse. Both men and women are actually to the same extent getting worse over time. And that's within the 24-month period. And that's statistically also significant. And the same we can see where you ask people to have their feet apart and close their eyes. And you can see that men are significantly worse here than women, but again, both of them are getting worse over a 24 months period. So this looks to be a pretty good outcome measure. One of the things that we're still trying to answer, and we're working with the Dutch group that now has given us some of this data from their side, we're analyzing, is whether these correlate with an increased level of falls. We think so, but we actually have to demonstrate that so that we can sort of pitch it to the FDA as an outcome. Another variable that might be very useful is measuring hip strength. So the hip flexors, the muscles that help you to bend your hip upwards towards your face, you can measure that with a dynamometer. And again, we've shown, this is work done again by Kathy and Jen Keller, that the more severe your neurologic disability is, the higher your neurological disability score is, the weaker are you in that muscle. And so I think it's actually a very sensitive measure. In the clinic, we see that sometimes one of the first signs is that the hip muscles seem to be weaker if you measure the strength there. And again, that there is a slight decline. It's significant, but it's not that dramatic over a 24 months period. So it's something that we think we should collect, but it's not as significantly different as the sway amplitude. So more recently, we have started measuring these things at home, because one of the nice things is that the sway amplitude we can also measure in somebody's home by putting on some variables. So we have these variables. They look like Fitbits. You can put them around the belly and you can put them on the feet. And with the sensor, and Amina has been really spearheading this. She ships all this equipment to the patient's home, and then we set it all up there. We'll measure their sway, we'll measure their walking speed, and then they send the device back to us. And we have now a handful of ALD patients, and then about a dozen of patients with another leukodystrophy. I think she presented some of the data already last year, where we see that there's a pretty good correlation between what you can do at home and what you can do at lab. So this could be potentially something where we can measure people's balance issues even at home without having to bring them in on site, every 3 months or every 6 months. So I want to pivot a little bit to other outcome measures. So two other categories that need to be discussed. One is imaging. Of course, we are talking about a neurologic disease where there is something wrong with the nervous system, and we can image the nervous system. So there has been a long-standing focus on imaging. This is how I got to work in ALD when I first joined Hugo Moser and Jay Raymond's group back in 2001, when I didn't have gray hair. Anyway, at that time, we we did a lot what we call MR spectroscopy. So this is a methodology to look at chemicals in the brain in a living person. It's basically like an MRI, and you can measure these peaks. And so each of these peaks is a reflection of a different chemical. And there is a chemical called NAA, and there's a chemical that's called choline. So NAA is thought to be something that is important for axonal integrity, so the integrity of the nerve cables. The choline is thought to be a reflection of myelination. And so when you look at the NAA to creatine ratio, which is a reflection of how much axonal integrity you have, this is this plot here, it did correlate with neurological disability scores. But there's been a lot of issues with MRI and MR spectroscopy in particular in terms of standardization. There are now in the last few years, a lot of efforts on standardizing this technique, but it's not that easy. You can't just go to any radiologist and get an MR spectroscopy. But that's not something that we can quantitate. We've been considering to use this as an outcome measure at the very least in trials for cerebral ALD. At least that's what I would like to do. Some companies agree with that. It requires some additional resources, and it's not very easy. Another imaging method that's been used a lot for the last 15 years is called diffusion tensor imaging. And there's several studies in ALD with DTI, including our own group, but I actually think again that Mark's group has published the most recent and most interesting results. They looked at, so with this method, we can look at sort of integrity of pathways within the nervous system. And they looked at the spinal cord here, and they saw a very nice correlation between the MRI signal and the neurologic disability score, the EDSS again, also an inverse correlation with the timed up and go test. But really what was interesting is that it looks like at least one of these indices called radial diffusivity changes over the course of 2 years. So this is a baseline, this is 1 year, this is 2-year follow-up. And so we see a nice increase over 2 years here. So this might be a good surrogate measure of outcome. But again, there are also here issues with standardization. And we've had one of my postdocs in the lab, Bella Turk, spending a lot of time traveling between different sites before COVID, and now I guess remoting between the different sites, trying to get standardized imaging sequences set up. And we all are working basically on those centers where we use the same brands of scanners, which complicates things to some degree. But none of these are really perfect. There is another method that I was involved with many years ago called magnetization transfer MRI. This is a really sensitive sequence to look at the myelin signal. And we had shown back then that in men with AMN and in women with ALD, there were both significantly increased signal in the dorsal column that they could detect compared to controls. And that significantly increased signal correlated more or less with the sway amplitude again, that sway test, and with the vibration sensation in the toe. So this imaging sequence was then subsequently used in that Lorenz-Azol trial that I just mentioned. Unfortunately, that's part of the problem with imaging, is that people upgrade their scanners and upgrade the software. So this was a multi-year trial where the scanners were upgraded to more modern scanners, making it incredibly difficult to correlate data. So we had one of our postdocs, Bella Turk, who's I think listening here, spending a lot of time learning machine learning tools and coding, trying to develop scripts that would allow us to make these sequences comparable with each other. And this is just a demonstration on how these sort of different analysis methods change the sensitivity of detecting differences. So here is like a more rudimentary type of analysis, and here is a more sophisticated type of analysis of the same images. And you can see how you can sort of split out differences here that you can't really see well on the first place. And so using these methods, he sees in that Lorenz-Azol data set somewhat similar finding than in the DTI studies that were done in the Netherlands, where again, we see a change in the magnetization transfer signal within a couple of years. So between baseline and 2 years, we see a significant change. It's not dramatic, it's not a huge change, it's a subtle change, but it's significant. And it's really just mostly in men. It's not really present in the women. In the women, it didn't really change that much, that signal. And again, there was, if you look at the change of the MRI signal of this magnetization transfer signal versus change in neurological disability, we do see a nice correlation and it's significant in males. It's not significant in women, but that implies that this imaging is changing over time. And again, it could be potentially used as a surrogate measure of outcome. We also have looked at spinal cord atrophy, and we see, as was also reported by the Dutch, that there is significant atrophy in the spinal cord. It seems to be pretty pronounced already at baseline, and it's more severe in men than in women, but also there isn't a dramatic change over 2 years again. So it's not a great, again, not a great measure of change. So not great overall. So then again, there's another category of biomarkers that we've been looking at, just looking at blood biomarkers. And there is one that is a hot topic right now in neurology in general called NFL, and it has nothing to do with football. It's a neurofilament light chain. It's something that we can measure in the blood, it can measure in the spinal fluid. The most comprehensive study for NFL in AMN was done again by Mark. And so in this paper, they did show a beautiful correlation between NFL and the severity score. So, you know, the more severe you are neurologically, the higher is your NFL value in blood. The challenge was that when they looked at it over 2 years, there wasn't really a dramatic change over 2 years. So it seems to be a sensitive marker that's abnormal, but it doesn't seem like it's really getting a lot worse in AMN over a time period of 2 years. Now, could it be that when you treat somebody, this value could go down? That's possible, and that's something that we were wondering. So we have looked with a group of folks in Michigan at other markers, more in a fishing type of experiment where we just sort of blindly looking at hundreds and sometimes thousands of different variables to see if we can find something. And so this was a pilot study with a small sample size. So we definitely are looking forward to repeat this study and trying to get a grant on this, although it's been difficult. But when we take controls and compare them to mild AMN versus severe AMN, we see that they fall in 3 different groups when we look at all the metabolites as a group, suggesting that these metabolites might be able to at least determine the severity of the disease. So then we looked at it in more detail. You know, there's hundreds of metabolites that we looked at, and we identified out of these hundreds of metabolites, a handful of metabolites that really seemed to be actually making sense. They were different in AMN, and they seem to correlate with some of the measures that we were looking at. And so when I carefully then zoom in there, we nailed it down to basically these 7 metabolites that were different, really different. And from them, there were really 2 that were quite promising because they were way different than the normal range. So these are normal ranges for these metabolites, and these are our patients. And there seemed to be that, at least with hypoxanthine, the more severe your disease is, the higher was the concentration of the metabolite. With 7-HOCA, it looked to be the other way around. And we suspect that this variable may actually be a predictor of whether you are going to develop cerebral ALD or not, but we can't prove that yet. So these are the 2 metabolites that came out of this pilot study to be interesting, and we're trying to pursue further. But now we can take all these metabolites and throw them into a machine learning tool. And with a little bit of magic and a lot of coding, when Bella looks at this data set, he can actually develop a machine learning tool that comes out and is able to spit with 83% accuracy what the neurological disability of that patient looks like just based on this metabolic signature. So it's not referencing a single metabolite or even a group. It's just if you look at the data overall, and that's sort of the power of machine learning. So I've told you a lot about these different outcome measures, blood markers, MRI markers, clinical outcome measures, and all of them are sort of imperfect. Each of them change a little bit. Nothing is like great, holy great type of approach. The question is, if you bring them all into a machine learning tool and combine them all together, can we then actually come up with a really good prediction tool that would not only predict how severe you are today, but how severe are you going to be affected in a couple of years? And so we would have to validate that, of course, in a large data set, but we're working on that right now with the pre-existing data set and trying to get grant funding to look at this prospectively in the ongoing sort of natural history studies that are already happening as we speak. So I want to just finish up thanking the group that's really behind this. Our center has gotten pretty big in terms of number of people. So it's actually 2 pages. This is the part of the clinical team. You know, many of these faces, you know, Kim has been with us for many, many years. You know, of course, all of you know Dr. Raymond. And we have a bunch of other people who are very involved. And it's 2 pages of people that I have to show. And of course, herself is still working and is probably in the lab right now listening. Wanted to also thank the outside collaborators. And some of this work was really not possible without NIH funding and Brian's Hope and Run for ALD and a bunch of other organizations that are really helping us. Thank you.
Thank you so much, Dr. Fatemi. It brings a lot of hope to a parent like myself, and I know others, to see that all this research is being done for AMN. There are a few questions in the chat. So I'll start with one from Sylvia. When my son was transplanted for cerebral ALD 17 years ago, we were told that if he survived, it was unknown if he would develop AMN later on. Have you seen patients with cerebral ALD who survived transplant cross over to AMN? Is evaluating for AMN recommended after transplant for the cerebral form?
I have personally not seen patients developing AMN, but the problem is a problem of sample size. There is a paper that was published by the Dutch group of 5 individuals who developed AMN after being transplanted as a child. I don't know if Dr. Raymond is on. I had asked this question like 100 times before, and he had not seen any yet, but that might have changed since the last time I asked him that question. It's the part of a problem is that you know, you need to wait long enough, right? And so many of these successful transplants have really just happened within the last 15 to 20 years. And as I said, the problem is we see patients not being symptomatic until their 50s and 60s sometimes. So it makes it a little difficult to really know what the answer is. But again, there is at least one publication out there. And I see Katie Baker just says she had at least one, right? So you know, it would probably make sense that it may lessen the severity of the disease, but if it's able to completely eliminate the disease is to be honest, unlikely, I think, from a mechanism. It's also that you as a transplant, you know, especially the older individuals, they had, you know, more significant chemotherapy that could be in itself, you know, deleterious to the axons in the long term. So also it depends on how much complications they had throughout that transplant process. So I think that the answer is not clear, but there is at least some data out there that yes, you still have the chance of developing AMN. Although it may be decreased.
At least might make sense to answer Sylvia's question.
I think Stefan has a comment.
Yes.
Yeah, I saw silent. The host wants me to unmute. Yeah, thank you for your presentation, Ali. Yeah, I would like to have one comment on the bone marrow transplant and late onset of possibly of myelopathy. It's true there are just a few cases have been investigated. I think my hypothesis is that they still develop it is because the bone marrow transplant takes away the inflammatory response in the brain, but it doesn't lower very long chain fatty acids. So our working hypothesis is that the myelopathy is caused by a chronic toxic effect of the very long chain fatty acids, which remain high. And unless we have disproven that hypothesis, we continue. And that's also one of the rationales behind AAV gene therapy in the spinal cord and other approaches for spinal cord disease.
Right. I think it's important, Katie made another comment, which is, I think, really important, is that we do hope that these boys would have possibility of AMN treatments in the future. So I think none of the therapeutics so far that have been explored would exclude them from being you know, bone marrow transplant will not exclude them from participation in those trials, I would think, in the future. Now, at least once these drugs are approved, so maybe at the trial stage, it might be complicated, but post-trial stage, I don't see any reason why we wouldn't want to treat individuals.
Okay. Lisa, I see you question. Oh, sorry.
No, I was going to say, this is Paul Orchard. So, yeah, we haven't seen any kids that we've been convinced have gotten a lot of AMN symptoms, but as Zali's pointed out and whatnot, there aren't that many kids that were transplanted that are in their, for instance, 30s to be able to sort these things out. But I don't know, Ali, do you know if the murine studies, I mean, the mice don't get cerebral disease, but they get something sort of akin to AMN, right? So, and at least from what I've heard, that some of the mice that have been transplanted, I don't know if I ever saw this published, but I'd kind of heard that some of these mice that were transplanted seem to do a bit better and not necessarily have the same progression. Do you know anything about that specifically? Or
I do not know about that, actually, no.
Okay. I only know of a recent paper from the Japanese group in which they investigated the effect of bone marrow transplant on variant chain fatty acids, and it didn't show any effect.
Okay.
But I don't think there's any clinical data on these mice in that paper. That's a paper from last year.
All right. Yeah, because I mean, it doesn't seem like transplant is going to alleviate, or gene therapy potentially is going to alleviate AMN completely, but whether it changes the onset of disease or the progression of the disease or whatever, I mean, that may take us another 10, 15 years to sort out. But
I think the comment here is that you buy a lot of time. And I mean, the field is moving very fast. So,
right.
So I think that's a very important point. And yeah, it may result in a lower or slower disease course. We don't know, but as Ali said, there's not enough follow-up cases.
Elisa.
Yes, so I had a question, and I think there was a study done a couple of years ago with exercise in women. So is there any proof that exercising and being physically fit will actually slow the progression?
So I actually, did she just sign off? I thought she was there. Anyway, I guess she signed off. So Jen Keller was here. She might have signed off, but she did that study. And yes, it was an interesting situation. So a large percentage of women improved in their hip strength as part of at the end of that study, which was a 6 months long study. Interestingly, those individuals who improved their hip strength, they also walked better, and they reported a better quality of life. But we don't know why only certain people did that, and whether that had to do with how stringent they stuck to their exercise regimen, or there was something biological on those individuals. We also don't have follow-up data since that study to see if those people are doing better or not. So I am following a bunch of those individuals in our clinic, and so does Amina and Dr. Raymond, and I mean, we certainly see people getting worse now. So, I don't think that it would have a long-lasting effect. It was probably more an effect on the muscles themselves, and strengthening weak muscles makes a lot of sense. So we do recommend exercises, especially hip strengthening exercises. That's something that we recommend, but whether it's fully sort of having a long-term effect or not, we don't know. And Amina just made a comment. We don't have IRB approval yet, but we're just about to start another exercise study for men and women this time. And the idea is that we would actually try to do that potentially partially online now with Zoom and the wearables and all those things that we have. So just stay tuned. It's not approved yet, but hopefully we can get there very soon. And it would be, I think, it's a short-term home-based study where you basically just dial in, give you instructions how to do it. You do it at home, and then we do a pre and post assessment of these same measures that I mentioned.
Yeah, if you could share that once it's available, I would love to.
Yeah, definitely. Once we have IRB approval, we might need the foundation's helps to spread the message anyway. So yeah, definitely.
Thank you.
Happy to do that. We're excited about getting started hopefully soon.
Thank you.
Just
Okay.
one quick comment related. There was a question, I think, about participation. So we have a study right now that's recruiting called the Global Leukodystrophy Initiative, which is an NIH-funded study with several sites. The sites include CHOP, MGH, Kennedy Krieger, Stanford, Utah Children's Hospital, and Emory and Baylor. And people can sign up at these sites. They may be referred to us for a particular quantitative sort of project where we are measuring these quantitative measures and the variable stuff. But they can all sign up for this overall study at these sites, or they can reach out to us. I can provide maybe our it's actually leukodystrophy@kennedykrieger.org. So that's probably the easiest, or my last name @kennedykrieger.org. So
you can share that. I'll send it to the group.
Yeah,
that would
sure.
be great.
Sure.
Thank you.